Hello there, I'm Ben, and today I'm going to give you a brief history of human genomics from when I was growing up as a lad till about today. The goal for this whirlwind tour is to really describe the purpose and objectives from my point of view of what the human genomics activity is all about, trying to discover genetic variation associated with complex traits. I'll give you a bit of history, at least from my perspective, about how we got here. Thematically, I'm going to try to couch this in terms of questions and models, but also data and methods that are relevant that then lead to, as any good scientific experiment, additional questions that remain to be answered after well-formulated ones are addressed. At the end, I hope to give you a little bit of the state-of-the-art directions as I see them in the next 5 to 10 years, but that might be for a second video sort of at the end. I'm a human geneticist, and my focus is thinking about and studying humans as a central agent. It turns out that it's actually really hard to study people relative to other model systems like a mouse. So why are we even bothering doing this? There are a lot of really great reasons for this. One main reason is that by studying humans directly, we really get to understand the genetic basis and the genetic architecture of disease and those traits directly in humans and across human populations. Every particular region of the genome we find in humans is itself a root cause of disease because somehow, some way in people, that small genetic variation, that change that has a small impact on the phenotype ultimately relates to some mechanism and some gene that relates to disease. So every one of those pieces of the puzzle we can put together tells us a larger story about the causes of disease. Ultimately, we're in the business of trying to understand and infer biological mechanism. And the idea that we could learn everything from studying a model system is a little bit simplistic. There are almost certainly complexities in the human system and uncertainties and new knowledge that we can extract from studying humans. And so by doing that, we can actually get to those root mechanisms. One day, at some point, we're going to also want to understand who is at most risk of disease or who has the lowest risk of disease. And so we'll start to think about genetic risk prediction problems. And so we ultimately, at the bottom line, want to do that in humans. And the only way we're going to learn those models and develop them best from genetics is by studying humans. And ultimately, one day, we'd really like to use these root causes of disease to understand how we can better treat disease. So if we find a gene that is broken and we can repair it somehow pharmacologically, the human genetic information that we use to make those inferences are actually really direct and can help catapult and jumpstart therapeutic interventions as they become available. So studying humans is hard, but one thing we really have to ask ourselves before we start thinking, oh, okay, I have this new trait and I want to dissect it, is that trait actually genetically, does it have a genetic basis or not? And so we immediately start thinking about families and this sort of like intuitive idea that we all have had where we have families and we know within our family we have particular predispositions for disease or particular traits that are unusual in our own families that we know pass from generation to generation. This is sort of very commonsensical and very intuitively known. This like heritability of traits is something we all kind of commonly understand. So whether or not a trait is genetic is really directly related to that familial aggregation of a trait or disease. So if you find a pedigree like we have described here, this is this 3-generation pedigree. And so the current generation has quite a few number of affected probands. But if you trace back in the most recent history, there are parents of those probands that were also affected, and even further back in time, their grandparents of those probands were also affected. One of those probands were also affected too. So this seems very plausible, but we also have to remember that confounding potentially the inference of heritability is this idea of shared environment. Mostly we intuitively understand that our brothers and sisters, we were all kind of raised in the same place. relatively close to the same time. So we probably had the same environment, the same exposures, the same kind of rearing. And so if a trait was caused by environmental exposures that we were just exposed to in common, shared those things, that might potentially confound our inference of heritability. However, we can address this with particular kinds of clever designs like twin-based studies. And I leave that to you, the reader, to think this, the viewer to think this through a little bit. Are there designs between twins, monozygotic twins versus dizygotic twins, raised in different or separate environments that might be able to hone in on traits being heritable or not? So once we start understanding the idea that like heritability is a thing that we can actually not just commonsensically understand, but we actually have this idea that not all traits have the same amount of heritability to them. Height, for example, feels very heritable because we kind of have this idea that our parents, if they're both pretty tall, the kids they have are probably going to be relatively taller, some kind of blend between them. But maybe a trait like measles may not actually be as heritable as we think, even though it's common. It might be common, or at least it was common many, many generations ago. So we have this idea that maybe different traits actually have different heritabilities. And if we could only start building a model that we could use to understand that trait heritability, it might give us a clue as to what traits might be more genetically basis versus less, and maybe more tractable to genetic dissection versus other traits. So this is an idea that started to be developed, tens to hundreds of years, decades ago to 100 years ago with some basis. And so we start to describe phenotypic variability in a population. So let's imagine we have a trait P, and we want to describe variance in that trait, the total phenotypic variance that is contributed by all sorts of factors. So it turns out variance, based on very, very first principle statistics, a variable can be just the variance in a variable can be described by the sum of different variances. So for example, the total phenotypic variance here is the sum of the genetic variance plus the sum of the variance contributed by the environment. And then any covariance that happens to occur between genetics and environment, which we know definitely can happen. Let's just say for the moment, to make things a little bit easier, let's assume that this covariance between genes and environment is actually 0. And we can imagine that there are many cases where that is definitely not true, but many cases where that's absolutely true. Okay, but for the moment, for simplicity, let's imagine that there were no gene-environment interactions. Then what we can actually do is quantify what's called broad sense heritability. And that just turns out to be the proportion of genetics that contributes to the total phenotypic heritability. So all of the genetic components that might contribute to variability, that's heritability, the proportion of that contributes to the total phenotypic variability. Intuitively, though, we understand based on sort of like first principles genetics back into our days of taking Mendelian genetics, that we actually know that genetics can take on actually many forms. You can have additive, dominance interactions between genes. So lots of components that could influence that genetic part. Again, let's for simplicity say that we're going to ignore all the other effects except the very straightforward additive effects. So if you have 0 alleles, you have no addition to the trait. If you have 1 allele, you add 1 unit to the trait. And if you have 2 alleles, you add 2 units to the trait. So very, very straightforward additive models, and there's no other effects. If we had that situation, we could define what we call narrow sense heritability. So again, very simplistically, that heritability is now, because it's only based on additive terms, the additive variance components to genetics over the total phenotypic variability, that contributes to narrow sense heritability. And it turns out this narrow sense heritability is actually intuitively really directly related to how we can understand and dissect complex traits in general, because for the most part, these genetic effects tend to be mostly additive, maybe a little bit dominant. They're not strictly Mendelian in the way that we normally understand monogenic disease-causing variants. They're more additive, and so you have these many effects from these tiny individual genetic effects that when they add up all together really contribute to a lot of phenotypic variability. So this heritability that's based on additive genetics turns out to be really, really valuable and important. And it turns out that we can use different types of familial pairings to actually quantify heritability. And the reason that we can do that, if we think about it just a little bit, is that the relationship between two different pairs of individuals and how many alleles they might expect to share identically by descent. So how many alleles are passed down from mom and dad to proband 1 to proband 2, given that how many they're expected to share. So for example, monozygotic twins will, for the alleles that are configurations where the alleles are shared, one allele is shared between them, that happens with probability 1. Dizygotic twins or sibs share 1/2. Half sibs share 1/4, and so on and so forth. So different configurations actually expect different patterns of identity by descent sharing. And so we can use over many, many different configurations, dozens or hundreds of monozygotic twins, which have a particular IBD pattern, or dizygotic twins or half sibs, to actually set an expectation for how genetic a component is. If a trait or a component is very, very genetic, then the proportion that the phenotype is expressed is proportional to how much identity by descent sharing those two probands have in common. If they both share the variant that expresses the trait, they're going to more often have the trait together. And so we can use the power of statistics and aggregation to make good guesses about what we think that heritability is, complex trait to complex trait. So these are experiments that have been done. I have some papers cited below that kind of gives an estimate from this sort of like classic way of estimating heritability. And it turns out that there are a lot of traits very commonly that are actually quite heritable. For example, height. It's like our stature is amongst one of the most heritable traits at about 70 to 80% of the variability that we see in a population of stature can be explained by additive genetic components to that total phenotypic heritability, which is incredible. Other traits actually are similarly heritable, but maybe not as much as height. So body mass index, for example, obesity, we conventionally think of that trait as behaviorally driven and an environmental exposure even perhaps. But it turns out actually obesity, as measured by body mass index, is actually quite heritable, between 20 and 50% heritable, which is also pretty incredible. Low density lipoprotein cholesterol, so that important risk factor for heart disease, where you're supposed to keep that as low as possible. And we have therapeutics that target LDL lowering. Even LDL cholesterol is heritable. Type 2 diabetes, not surprisingly, is also heritable, around 20 to 40%. A trait, neuropsychiatric trait like schizophrenia, turns out to be incredibly heritable, 60, 80%. So in practice, many of the complex traits that seem exceedingly complex turn out to have a great genetic basis to them. And so, having these quantifications of heritability for these traits sort of opens the door to other questions. Is a trait heritable? If a trait is heritable, can we dissect it genetically? And if so, how are we going to do it? Once we start thinking about the components of heritability and a trait, we start to realize that there's actually quite a complicated architecture that you could use to describe that component of additive genetic variants. And there are a lot of different concepts to think about here. And these are by no means all of those concepts, but I think these form kind of the basis of many of them that we commonly think about when we think about the complexity of a trait. And again, these are nicely reviewed in this very recent Nature Reviews Genetics article that came out a couple of years ago. But let me just kind of like enumerate them specifically, because I think this is kind of interesting to think about as we're imagining sort of like how we approach dissecting traits today, but also in the past, what people thought about these particular forms of architecture and how that's evolved over time and how that's changed our models and methods and data that we've used to try to dissect them. So probably the most important one is genetic heterogeneity. And this is simply thinking about the number of distinctive genetic loci that actually contribute to the trait. So a simple way to think about that would be You've got gene A that's on chromosome 1, and then you've got gene B on chromosome 2, and maybe a gene C on chromosome 3. And they are on separate chromosomes. They assort independently of each other, but maybe each one of those separate, distinct loci have are risk factors for a particular trait. So mutations or polymorphisms that are in that gene might actually influence the trait somehow. And in this case, in this very simple model, there's like 3 of them. And so you can imagine there's actually on the range of complex traits to simple traits, you could imagine many, many different loci to very, very few loci contributing. So for example, in classic architecture, a monogenic trait only has a single gene that contributes to risk. So if you see a particular rare phenotype, perhaps it's only contributed by one gene breaking. And when that one gene breaks, that causes the disease. Those architectures are much simpler to dissect than, let's say, an architecture for, let's say, height or schizophrenia, where maybe it's not one gene, it may not be 10 genes, it's probably not 100 genes, but maybe it's thousands of genes, thousands of distinctive physical locations in the genome that matter. Genetic heterogeneity is subtly different than, let's say, allelic heterogeneity. So given a particular locus or gene, this is referring to the idea that there are a number of variants at that locus that could influence the trait. And if we for a moment took off our human genetics cap and put on our model systems cap, this would actually make a lot of sense to us. And we commonly in model systems biology think about multiple mutations in a gene that influence the trait. And I could cite a number of classic examples. My favorite of them is fruit fly eye color. There's gene, the eyeless color gene, where you get mutations in that gene, and you can have the range and gambit of fruit fly eye colors from red to white and a lot of things in between. And those are all caused by just coding mutations in different spots in the gene that's responsible for that. So you can get at a particular physical region or a particular gene, you could have different types of mutations that are all independent of each other or rare or something that somehow contribute to the trait. That's what we think about when we think about allelic heterogeneity. Now, again, intuitively, implicitly in this, I've sort of made this reference to effect sizes. So, for a given mutation that is influencing the trait, those particular lesions may have a small effect, a medium effect, or a large effect. So when we think about the effect size, again, we're thinking about this in this sort of like additive model. We're thinking about what is the direct relationship between change in genotype to change in phenotype. So as you increase the dosage of that particular allele that we know is causal, what is the downstream impact on the phenotype of interest? And typically, again, we're thinking about these additive models. So we have some intermediate genotype and we measure the relationship between loss or gain of an allele, and then we quantify the change in that phenotype, or the additive effect here in this graph below is A, and that estimated A is in fact quite proportional to the effect size that we might estimate when we do simple regression models to estimate the relationship between genotype dosage and the phenotype of interest. So we've got the number of locations in the genome for a given location of the genome, the number of alleles that influence the trait, and then for each allele, we have the impact of that allele on the trait. But we also have frequencies of those particular alleles in the population. Again, we're typically thinking about this not just with a single patient in mind, we're thinking about this at the population level. And so a particular variant may be exceedingly rare, all the way to being de novo, only seen in that individual and never seen outside the population, to variation that may be actually quite commonly observed in many, many individuals in the population because it occurred tens of thousands of generations ago, and has sort of sojourned and had been, has maintained its frequency and is actually quite common in the human population. So we have the number of loci, the number of signals or alleles at a locus. You have the effect that that variant has on the phenotype. You have how prevalent that signal or allele is. And then potentially across different alleles or different sites in the genome, you could potentially have interactions. So non-additive effects across multiple alleles that may modify the phenotypic value or the disease penetrance can also happen. And, all of these, these don't count all of the possible components of architecture, but when we think about architecture, we're generally referring to many, if not all of these components in addition to others. It's interesting to think about the relationship between some of these components of architecture. And a lot of this comes from, I think, thinking about, you know, thinking about this model from both a statistical point of view and a population genetics point of view. So what you can see on the plot that I've shown here is sort of like a cartoonish version of thinking about this model, because at some level, we know intuitively there's got to be some constraints on the relationship between the total prevalence or number of alleles, their effect, their frequency, and the total prevalence of a phenotype in a population. So if a phenotype is exceedingly rare, then, for example, it's statistically impossible to have that variant be super common and perfectly penetrant, because if it was, then the frequency that we would observe, the prevalence of that trait would be very, very common in the population. But empirically, what we intuitively know is that Mendelian mutations, those monogenic forms that are quite rare, are observed very, very infrequently, and they're highly penetrant. So this, on the left side hand of the graph, we see like rare variants that are, direct causes of fully penetrant monogenic disease or nearly perfectly penetrant. And as you increase in frequency and have increasingly more variation contributing, where you get more complexity, the effect sizes tend to just be attenuated because you have more phenotypic possibilities, more potential to influence the trait, and those things don't occur very much. That's sort of like statistically what we see. And if you actually, this is a theoretical example, sort of like cartoonish on the left, but if you actually go to data, I've pulled together, for example, this plot from a recent genome-wide association study where they mapped genetic variation for cholesterol. And what you can see is actually this in practice. So if you imagine the distribution of the trait increasing in frequency, you can see that as the frequency of the variant gets super high or super low, i.e. the minor allele is, in both cases are actually quite low, you can see the effect size actually gets quite potent. And part of that is a little bit of statistics, and a part of that is population genetics. So let's think about this just for a minute. Why would this distribution be the way that it is, right? Like, why in practice do we observe rare alleles of strong effect and common variants of weak effect, but not rare variants with weak effect or common variants with like super high penetrance effect, okay? If you think about it just for a little bit, you can imagine two sort of like forces that might be at play. Like one's like practical biological and one's like practical statistical. So on the practical statistical side, rare variants that are pretty weak, we're probably just not, we're probably not going to see that variant very often. And when we do see it, it doesn't have very big of an impact on the trait. And so statistically, the chance that we're going to actually be able to discover it is probably pretty weak. On the other hand, common variants that are actually pretty strong, if they were phenotypically consequential, maybe even like evolutionarily consequential, like, breaking a gene changes your trait by a lot, but it also may have some impacts on your evolutionary fitness, then background selection starts to happen. And sort of like over a very, very long period of time, minor effects that reduce your fitness are going to gradually be purified and pushed out of the population. So what you have left is ultimately variation that's sort of like tolerated enough by selection that you can actually see it as polymorphic in the population. So it sort of constrains what you actually observe in practice between what we ultimately see, some clinically relevant trait, but then it's sort of like attached to some hidden selection effect that we don't know about in evolutionary terms, but we can see the imprint of that effect because we know the phenotypic value is constrained, and the effect size is constrained, and the allele frequency is common as a result. So between these two forces, we can see how we might not see things on one side, but then also not see things on the other side as a result. So if you start thinking about this, you start to imagine like, okay, well, what designs actually are better able to capture different architectures? So you told me, earlier you said, if a trait's heritable, then it can be genetically dissected. And we learned that from family-based studies, but it seems like there's like lots of different components of this like genetic architecture that may be easier to dissect versus others. Like, for example, if a gene if a trait was caused by a single gene, then you could imagine that maybe one type of design might be able to dissect that much better than a second trait where, perhaps the gene, there's like 1,000 genes or 1,000 loci in the genome that matter for the trait. So what's going on here? So this intuition is actually not wrong. It's quite reasonable. Not all the experiment designs that you could possibly imagine are going to be perfectly powered to dissect all of the aspects of genetic architecture. And I'll start to unpack for you a little bit. It turns out historically we started thinking about this based on data. We use data to try to drive our inferences about what we think might be going on. Built experimental designs to try to tackle what we thought was going on. Realized that those weren't working as well, and then had to like change tack and try different designs that then were much more successful subsequently. So really starting this off are a long and deep history about monogenic and Mendelian diseases. And there are lots of great examples here. For the purposes of this discussion, I can't help but pick a trait set that's closer to my heart, which is familial hypercholesterolemia, or FH. And this is a trait that we've known about for, a very, very, very long time. And, probably reported in a very, very specific way with these particular xanthomata that sort of like in patients with FH appear very, very robustly. And if you see this picture, you can see right away it is a very distinctive phenotype. You can recognize it almost immediately. And the physical manifestation here is sort of annoying, but like not life threatening. But it turns out that it was exceedingly well known that patients that had this particular manifestation physically often very suddenly would die of a heart attack or heart failure very, very, very early in life. People at this early stages, when they were aware of this phenotype, did what we would all do. We started to talk about one's family history, and it became immediately clear that the pattern of inheritance was autosomal dominant. Like if one of your parents had the trait, you were very, very likely to get the trait. If both your parents had it, then you were, very, very likely, and the patterns of inheritance sort of matched this autosomal dominant inheritance pattern. However, it was actually pretty rare, so not not like the 1, 5, 10, 15%, although if that were true, it would be terrible. This trait's rather rare, on the order of like 0.5% to 0.25%, somewhere in there. So we knew about this trait, and we knew of the disease manifestation, but it wasn't until sort of like in the more modern molecular biology and genetics era of the 70s and 80s that actually we started to do molecular genetic dissections. And it turns out that one of the main causes of AFH is actually due to mutations in the receptor of LDL cholesterol. And turns out that that is direct causes of disease. And all of the things that follow from that are, generated multiple Nobel Prizes, therapeutics that are exceedingly good at lowering LDL cholesterol and are the first-line defense of heart disease prevention, followed from that in subsequent years based on this observational data. So the promise of that is amazing. So like, we could go from like an observation in humans, an understanding of the basics of disease, using the tools of molecular biology to discover the risk factors, and then use that to springboard therapeutic development around that molecular mechanistic biology that we now understand, is a transformative idea. And it's sort of like why the power of human genetics is so gripping in terms of trying to follow things up. So based on this, in not just this example, but there are other examples I could have cited. So it's like, well, could we take advantage of this approach for other rare diseases and just start mapping the underlying genes and the molecular mechanism with the idea of like, we could use that to like create the next set of statins and cure schizophrenia, or reduce obesity and cure Alzheimer's disease. That would be amazing. So where do we start? Okay, so linkage mapping was sort of like the natural thing to think about, because this was a technology and an approach that that others had been using in other contexts very, very readily. And my favorite story about this is of course Alfred Sturtevant, who famously in 1911, I think he was an undergrad, and rather than do like his homework for the week or something, he was working and doing research. And so instead, he created the first genetic map in fruit flies to map the ordering of how genes kind of were ordered in subsequent. And based on just sort of like the frequency in which the traits and the genes underlying those traits were sort of like segregating through recombination, so parental versus non-parental phenotypic ordering, and realized that, you could actually use that. The frequency of those recombination events or those non-parental types give you a way to order the gene, and the frequency of how far apart or close by those were. It was a transformative idea, but didn't bother with his homework, just built the first linkage map, and ta-da, ushered us into a new era of sort of like mapping and ordering genes along a chromosome. So that intuition was brought formally in 1955 by Newton Morton, who basically took that intuition and converted into a statistical test, a test to determine whether or not loci are actually linked. And probably many of you have heard this. It's the so-called Lod score. And the key idea about the Lod score is trying to estimate theta. And theta is that recombination fraction between two loci. So intuitively, in this, like in Alfred Sturtevant, he was sort of measuring theta, the distance between two sites, by the frequency of parental versus non-parental phenotypes. So you get a non-parental allele if there's a recombination that breaks apart the genes. And so similarly here, theta tries to estimate what that recombination event is. And so basically you can calculate the statistic, which quantifies the chance that you see the phenotype or you don't, given the amount of recombination between them. And so what you might do is get really excited if you find that the trait and the marker you're using to tag to the trait are often segregating together. So they're often transmitted. There's not much distance recombination between those. And the best fit to the data is one where the marker is basically sitting right on top of, or is very, very close by to the trait you're interested in studying. And you can very, very estimate that effect with this statistic. And that can kind of give you a way of in family or single family or perhaps across families, estimate whether or not a variant is linked to and is transmitted with a particular phenotype. Okay, so this is grand. So like you could do this if you had a marker and a trait, or you had two traits and you were working in flies, and you could do that sort of linkage analysis. But like in humans, what are you going to do? I mean, you want to apply the linkage mapping framework to it at scale, but how do we actually go about doing this? And around the time that all these things were developed, our forebears in human genetics were imagining how you could actually do this and thinking about this in terms of a random set of genetic variants that kind of were distributed across the genome. So, enter David Botstein and others around the 1980s, thinking about the idea that actually, at the time, the only measurements of polymorphism that we had were these sort of like restriction fragment length polymorphisms. So basically, taking naked DNA and cutting it up with an enzyme and then measuring the lengths of those fragments that created on a gel, and then realizing that some people, when you do this, have a very different fragment length distribution than others, and then using that kind of readout to try to quantify individually across the genome, each one of those sites, to try to build this linkage map that you could use to then correlate with disease. So this paper kind of introduced this very, very basic idea that maybe what you could do is actually use extant polymorphisms like RFLPs or other types of markers to define this map and then sort of like map distances between those particular sites. And then once you had the distances between them, you could then layer in a phenotype of interest and try to connect the phenotype to that marker that's sort of like distributed all, one or more of those markers that are distributed throughout the genome. And you'd be really excited if you found that a particular marker was often found segregating with the trait of interest. That would allow you to sort of like map that site and figure out exactly where susceptibility to that trait might be occupying. And it turns out that approach worked really, really well. We spent the next 30 years or so identifying families that segregated these rare phenotypes, and we found a slew of genes that underlie those rare Mendelian diseases, and we built a catalog in OMIM and other sources of what those genes were and what phenotypes they were attached to. So this was an exceedingly important approach, and it worked exceedingly well. And it still works even to the relative modern era when I started to be a graduate student. Even for something that isn't perfectly Mendelian, but is actually less frequent, not super rare in the population, but not super common either. For example, type 1 diabetes turns out it's an autoimmune disease, and one of the main molecular players in defining the immune system is its ability to recognize and distinguish self from non-self. That happens at the human leukocyte antigen locus. That's the analog to the major histocompatibility complex, the MHC. So in humans, that's the HLA. That's the important site in the genome. And it turns out when you collect many, many families all segregating type 1 diabetes risk, you can apply the linkage mapping framework and identify that the HLA is a strongly linked region of the genome with that trait, a LOD score of 65.8. So a massive, massive signal telling you that like, this is a place in the genome that really matters for this trait. And you can find that important site with this approach. However, when we started to turn to complex traits beyond, more complex genetic traits from our rare monogenic forms, people were doing these experiments, and we weren't yielding the productivity that we thought we would expect, given the success in monogenic disease. And so the question started to be raised: why aren't we finding all the things that we're expecting to be finding? And, really, this was an active debate and discussion in the community. And Neil Reisch and Kathleen Merrick-Hongas published this very, very influential paper in the mid-90s, where they basically pointed out that, there are different types of architectures that may be more or less tractable with different types of experimental design. So on the one hand, we've been thinking a lot about, historically up to this point, about linkage studies. And if it turned out that the effects that we were trying to find for those particular loci were actually much weaker than we were hoping that they would be, the amount of family data that we would need to dissect those traits and discover them would start to become like astronomically infeasible for anyone to discover, let alone explaining why we haven't found any of those if the largest aggregation of family studies are in the order of 700 or 800 families, for reasonably strong effect sizes, but relatively modest by Mendelian standards. Effect size on the order of twofold or greater might require thousands, tens of thousands, maybe even hundreds of thousands of families to dissect. So this was a statistical argument, perhaps intuiting why linkage analysis isn't going to find everything, but it's going to find some set of things that definitely matter. They also lightly, delicately, reasonably suggested that perhaps other types of experimental designs might actually be more appropriate. So if you took a test for association instead of a test for linkage, with a similar type of experimental design with shared pedigrees, you might actually dramatically be able to improve the power for discovery of substantially much weaker effects. with that design in mind. And that is an incredibly potent an idea, and at the time a very controversial one, as we'll discuss. But a point still stands. So if we were to think about and try to embrace this idea of, okay, maybe actually testing for association, so the relationship between the allele dosage and the phenotype, not, not necessarily in a family context or a small affected SID pair design, or maybe even in an anonymous case control design. Let's say that we believe that that was the best design to sort of tackle identifying variation that is has an effect, but maybe not on the fully penetrant, much weaker effect size distribution. So we don't have the tools at the time of 2000 to do that experiment. What are the tools that we're going to need to actually do that? Well, prior to 2000, we have to remember we didn't actually have a reference genome for the human for humans yet. That didn't come along until 2000 and 2001. So, okay, prior to 2000, we didn't have it. But okay, after 2000, we actually have a human genome reference. So that was important because now we can start to say, where do we need to attach anonymous polymorphisms to start testing for association across the genome? Maybe we need something more, maybe we can get away with the density of a linkage map. Probably not. Probably we need something more dense, but we need to find all the sites in the genome that matter for genetic diversity as well. So we need a reference genome, then we need to map genetic variation and genetic diversity across human populations in that new reference genome. But we're still left with a question, how many markers do we actually need in the genome? In the linkage context, linkage mapping context, we could probably get by with diverse microsatellite markers or RFLPs that were very, very far apart from each other on the order of like a centimorgan apart, where you only expect one recombination event 1% of the time in a generation. So from the point of view of linkage, that gives you enough information to measure and estimate recombination well. But markers may, depending on how closely or how far apart or how tightly linked recombination happens at a very fine scale, will have a dramatic impact on how many markers we actually need. If recombination was frequent, then we were going to need a lot fewer, a lot more markers, because markers that are close by will be randomly shuffled more frequently. But if recombination is colder and more cool, then maybe we can get by with fewer markers further apart. And also, we just don't understand what common phenotypic variation looks like in general, and what our models for the architecture that is. Because we have very few examples that. We have a lot of great examples in the Mendelian case that we just now mapped for the last 30 years, but very few examples of complex trait signals being mapped where you can pin down a genetic variant and sort of use that as prior data to dissect what does that architecture look like, and what should we really be spending our time looking at. So it turns out all of these problems were recognized and ended up being solved at one level or another. So, in 2000, 2001, the human genome sequence was created. And then in parallel with that, an international group started to detect and discover single nucleotide polymorphisms that it could discover in that reference genome. So places where differences between U or I might be common enough that we can capture. And so started to collect and document all of the places where those single nucleotide polymorphisms occur in the genome. And this paper is sort of like the first large-scale analysis that describes that diversity in a very, very small number of people, but a very, very large number of polymorphic sites at the time. This is around circa 2001. Immediately recognizing, for example, that a lot of this variation is non-coding, not coding variation, even though you do find some coding variation. That's interesting in and of itself. But this was a paper really describing diversity. So it doesn't say anything about the relationship between genetic variants to one another. All it's saying is what and how frequent we see differences amongst people in individual populations. Okay, so in case you haven't thought about linkage disequilibrium or LD, have to give just a quick crash course in what linkage disequilibrium is. So LD just refers to the idea that alleles at two sites are assorting randomly or non-randomly with respect to one another. So let's say we have alleles A at B and B at two loci. what we can do is define the disequilibrium between them by calculating this simple quantity A capital B, which is simply the frequency of the gametes or frequency of haplotypes where the individual carries the capital A allele and the capital B allele at locus 1 and 2 times subtracted to the chance that you saw that given the the allele frequencies of A at locus 1 and B at locus 2 accordingly, right? So to give you some intuition about that, if the frequency of the haplotype carriers of capital A and capital B were the same as the frequency that you would see if those alleles are segregating independently, then D will be 0, and you could infer that the loci are in leakage equilibrium. There is 0 or low LD between them. However, if the haplotype frequency is quite high relative to the expectation based on the frequencies of those alleles, that would suggest that those alleles are actually not segregating randomly, that somehow the process of recombination is not acting, doesn't have a heavy hand in distinguishing or assorting the frequencies of the alleles between site 1 and site 2. If you think about this population genetically, we know that the frequency in which alleles at two different sites segregate will decay over time. And that decay is proportional to the recombination rate. So if two sites are very far apart, you have a lot of recombination between them, the linkage between them is going to go down precipitously fast. 1 generation, 2 generations, you're going to get random assortment between those two sites. Two sites are close together, that means that there's only a small window that recombination can operate in between those two sites. And so those two sites will tend to segregate their alleles together jointly, and only occasionally recombination will break them apart. But you have to remember, over evolutionary time, we're not talking about a single generation in a pedigree. We're thinking about a large number of recombinations over a large period of evolutionary time, tens, hundreds, thousands, tens of thousands, hundreds of thousands of generations. So it turns out that if you can model the LD decay as proportional to both recombination rate and the distance of time over generations. So the decay in LD is literally proportional to, we can model this as proportional to the recombination rate between them. An important related measure to this disequilibrium coefficient is what we refer to as R squared, which gives information about the prediction and ability to tag these variants. So this R squared value is simply, is equal to the square of the disequilibrium constant normalized by the allele frequencies of all the alleles at the site. So two sites that are segregating independently have a D of 0 and an R squared of 0, whereas with a disequilibrium constant that's high, where there's only actually 2 haplotypes in the population, R squared will be 1. And that effectively means that if you have the information of the allele at site 2 and you know that those two sites were perfectly linked, then you can perfectly predict what you guess the site is at the first site. Because you know those two sites are perfectly linked, you can say, oh, if I see the capital B allele, I know that at site 2, I know that the capital A allele must be present at site 1. So once we have this idea of linkage and linkage disequilibrium, and the proximity to how many markers we need across the genome comes up, because you now start to think, what's the most efficient design? Like, I've got 1.5 million single nucleotide polymorphisms I could genotype, but I probably don't want to genotype all of them. I probably want to genotype as few as possible so that I can actually capture all of the sort of like recombination free or recombination burden landscapes in the genome, to try to intuit where, to build the technology to actually ascertain this. And so again, around this time, and again, this, Lena Krugliak, again, a great thought leader, published this paper in 1999, before the human genome sequence was formally released. And because he was tied into thinking about the relationship between LD, linkage mapping, and use simulations to estimate under different models of linkage disequilibrium how many SNPs you might need to actually cover the whole genome given that model. And what he found purely based on theory and theoretical basic first principles modeling, suggests that maybe we needed upwards of like 500,000 SNPs in order to capture the genome variation as a whole. And that would account for different types of LD patterns that you might see in different populations that might have experienced different demographic histories. Population bottlenecks versus exponential growth might change the expectation of what you think those populations might have in terms of their patterns of linkage to equilibrium, because that influences how little or how much recombination history those populations and genomes might have experienced. A couple of years later, theory is great, but you really would like to have data to ground that theory. So what does the empirical data actually suggest about this? Well, in 2001, David Reich and colleagues generated some data to try to quantify what is the relationship between LD decay in the human genome. And this was only a small amount of data, 160 kb at relatively few sites in the genome, like 20 sites in the genome, in only 44 people. And what they discovered is that actually in these people and populations, that LD actually extends much further than they previously thought might. And really motivated this idea of like, well, maybe if it differs population to population, maybe we need to like survey empirically in different population groups patterns of linkage disequilibrium so that we can really better calibrate the association studies that we think we might want to gear up for, so we can design the most efficient experiments going forward. So as this idea of collecting and measuring differences in LD across populations started to become more in vogue, there was another conversation that was happening along the way, thinking about a different aspect of genetic architecture, where, for instance, thinking about that rare variant, high effect size, common variant versus weaker effect size discussion, suggesting that perhaps, unlike rare disease, which are predominantly driven by low frequency or rare variants, maybe even de novo variants, perhaps common, more prevalent disease is actually driven in large part by common genetic variation. And as a result, maybe what we actually need to do is capture patterns of common genetic variation and their patterns of LD concomitantly, so that ultimately we can do the linkage, the LD association mapping approach that we want, efficiently. If LD extends a long distance and we can capture common variation with a handful of genetic variants that tag that common haplotypic diversity, then in practice, we could design very, very efficient studies and technologies that capture that variation sporadically throughout the genome, that captures the common, most prevalent part of that variation, and then use that as a groundswell to test for the relationship between genotype and phenotype. And this is sort of like argued and motivated by sort of like the differences in the power differential between linkage analyses and association studies and the effect size estimates that we're talking about. Again, the technologies and the statistics and the data we had were calibrated to test a particular set of models, but not all possible models. So in the linkage case, low frequency variants with strong effect, where the nature of the disease is oligo or monogenic, this is where linkage analysis tends to have its best power. But for common variants with weaker effects, where the polygenicity of the trait is higher, this is perhaps where you need alternative methods. And this is where association studies were hypothesized to be the best powered approach to take these associations on. And so again, motivated by that idea, that's what led to, in part, the foundation of a project, a consortium, whose goal and design was to catalog genetic variation in multiple common haplotypic variation across multiple human populations in order to understand the nature and structure of patterns of linkage disequilibrium and common genetic variation across the genome. And this was wildly successful. So, this project, the HapMap project, surveyed 4 different population groups, mapping and genotyping 3 million genetic variants. It took 3, 4, 5 years to capture that variation, but it was a huge resource. It really elucidated population-specific patterns of LD is something that we need to really think hard about. And actually, as a corollary, though not directly related, led to this observation and this idea that actually haplotype blocks were sort of like punctate. There were sort of like these long stretches of haplotype blocks that were separated by a large amount of recombination that distinguished them. And what we learned later was actually that one way to think about modeling those data is actually that the landscape of recombination is not strictly uniformly distributed through the genome. It's actually distributed in punctate hotspots of recombination. And it turned out later, after a couple of years, model systems work, theoretical population genetics, thinking from a lot of great and very, very smart people, the discovery of PRDM9 as sort of like a mediator of hotspots and recombination in the genome. And that's a great set of, it's a great story, a great set of experiments, a great biological insight that wasn't designed to fall out of this experiment, but the data motivated the model and led to new discoveries that way. So again, after this sort of like large effort where we're trying to build tools, build resources, estimate, collect data, test our theoretical models, we now, at the end of about 2003 and 2004, actually finally have the basic reagents to start performing LD association mapping at scale. So, if you haven't thought about this, GWAS is like prevalent today, but I just want to like give a very, very quick overview of what experimental design is in practice. And basically, it's an incredibly straightforward design. Really, the goal here is, you're trying to do a correlation analysis. So you're thinking about a regression model where you're trying to predict a quantitative trait, or let's say you're trying to predict case control status. That's what you're trying to predict. And your hypothesis is that a genotype or genotype dosage at a given site in the genome is a predictor, correlates with that outcome. So change in genotype dosage is associated with change in phenotype. So what might you do? So what you would do is you would go, collect a bunch of polymorphic sites scattered throughout the genome. Either you've built a technology, an array technology that has selected what those variants are. So maybe you're not the one that's choosing them, but you have this sort of like large panel of genetic variation that you've surveyed. You apply that genotype, array to a large number of individuals who have a trait you're interested in. If in a disease state's a case control, but maybe it could be in a population of 1,000 or tens of thousands of people. You've measured their weight or height or LDL cholesterol. And really, you're just collecting sort of like those variants, but you know that those you're not genotyping all variants in the region. You're relying on linkage disequilibrium to sort of tag the haplotype structure so that either partially or perfectly, you're capturing a lot of hidden, either unobserved or, as we'll describe later, maybe imputed imputation guess genotypes locally in the region. So it's sort of like an efficient design. You have two SNPs that are in perfect LD. So one SNP tells you perfect information about the second SNP. You should only really bother genotyping the first SNP because you can learn exactly what the nature of the second SNP is. So in this study design, ideally you want to collect unrelated individuals in contrast to our family-based linkage studies where you're literally collecting pedigrees. Related individuals in this context are going to, are not independent observations. So in the regression analysis models, where you're assuming that all of the observation genotypes are sort of mutually independent of each other, if they're related, there's dependency structure in the data. And so that will change your understanding of what the variance properties of your test statistics are. So ideally, you want individuals to be unrelated. So the technology and the properties of this are kind of already laid out. How well you can capture different variation depends on the structure of the data in LD. In this very, very early days state, the idea was that we can capture common variation. So that's not going to capture all genetic variation across the frequency spectrum, but let's test this hypothesis and see if we can actually discover things. In collecting case control or phenotype data from a large cohort of unrelated individuals is a far better and easier study to design to collect data from. Collecting pedigree and family data is actually quite difficult and hard, and there's not a lot of data to necessarily do that. It requires a lot of time and effort and investment from epidemiologists to actually work with families to collect that data. But case control data, where it's unrelated, that's relatively easy to collect. You could go to a hospital system and ask people to participate in a study and actually make a lot of progress with collecting data. So then once you've collected that data, again, you're thinking about a regression model where the alleles you've typed at the genotype, let's say if you had an AG SNP, you've got dosage of the G allele. So that's either 0 copies, 1 copy, or 2 copy of that allele. So that's the variable that you're going to use in your prediction model and your regression model. So in this case, where the outcome Y is a quantitative trait like height or BMI, you can perform simple linear regression where you're regressing your genotype on your phenotype. And you're looking to test the hypothesis that effect size that you estimate, the slope of the genotype relationship, change in dosage to change in outcome, that beta effect size is either, is different from 0. And again, in this model, depending on the test statistic you have to do this analysis is a t-test distributed statistic, whereas, you get a pretty large number of observations that sort of like asymptotically trends towards normality. So this statistical framework is incredibly straightforward, very, very easy to implement, and very, very easy to apply. And the game that you're playing, of course, is you're not just testing one SNP in the genome, you're following that Leonid-Krugliak model, you're testing 100,000, 500,000, a million SNPs, maybe 2 million SNPs or more. Okay, so you might think at this point, well, that's pretty easy, Ben. Why did it take us so long to do this? Well, it turns out that actually there are complexities, and this kind of gets back to a lot of the difficulties that people had when the conversations were pretty vibrant, the debates that people had around, oh, we should do linkage analysis versus do something like association analysis. People were arguing about what the right biological and architectural model was, but at the same time, they were also arguing about ways in which the family-based designs are just generally a much more robust, healthy statistical analysis relative to the case control study design, which definitely has challenges with it. And so, for example, I've listed a couple of them here. I'll go through them very briefly. Talked about relatedness, right? Like if you have samples, 2 individuals that are related to each other, that sort of induces correlation between the observations of genotype and also phenotype. And so that will induce, that's not the expected distribution that the tests are assuming when you're applying them. And so that might require, if maybe you can invoke a study design where you intend not to collect related subjects, but we also appreciate that may not actually be possible, and that at some level, any large-scale data collection is going to identify relatives or related individuals, even if they're more far distantly related, like cousins or first cousins or second cousins. So that's one issue. Another issue to think about is just how you're genotyping the data. It's not as simple, these are early days in genotyping, and you've got array-based technology doing this. We're not living the sequencing era just yet. So, how are you generating the data? And of course, as we all know, data that's generated in batches and is not appropriately calibrated with respect to batch can very easily induce confounding and false positives. There are great examples of this in the literature, and it's an important issue to think about. To give you an idea about what the challenge could be, let's imagine a terrible design where you genotyped all of your cases on one day with one technician's hands, but then a different day in a different lab with a different technology, with a technician that was having a bad day genotyped all of your controls. So that could create a situation where you now could observe a difference in the genotypes and the frequency of particular genotypes at a SNP, but that could just simply be due to the way the data was generated in lab 1 versus lab 2, not having to do with actually a phenotype difference association. Now, there are, of course, designs where you could address that. So if you interleaved cases and controls in lab 1 and cases and controls in lab 2, then yes, there could be differences across labs, but you could in your design include that differences in lab protocol and timing in the analysis as a covariate, and that would kind of address that kind of effect. But batch of, we were aware of these effects, and like some of these things are not necessarily that obvious. So there was like great concern about how to think about and controlling for those effects. Probably the most important one out of all of these is actually this idea of population stratification, which is another confounding variable that could interleave between genotype and phenotype. So, if you can imagine a confounding model where you're trying to, identify a relationship between genotype and phenotype, if you have a tertiary variable, let's say population, a population ancestry that relates to both the genotype and the phenotype, then you could, without accounting for those differences, induce artificially a correlation between genotype and phenotype. And you might think, well, how could that happen? Well, in practice, that can happen very easily. So we know, for example, from population genetics, first principles, also empirically, that there are true differences in allele frequencies between population groups. That's a thing that we know and a thing we appreciate. We also know that the prevalence of disease is also potentially different between different population groups. And even if it wasn't different, you could imagine having the ascertainment of individuals across different ancestries to be different in a given design. So that would lead you to the arrow of between the confounding variable ancestry and the endpoint exposure, outcome that you want to test. But also we know intuitively that population label also correlates potentially with genotypes. So any place where there's a genotype that is different across populations will correlate with that difference between the ancestries that you might collect. And you might infer an association that doesn't have anything to do with true biological genotype phenotype. It simply has to do with the population collection you maintained. So that was a big deal. That was an extremely important issue. And it still is an important issue to think through. And people were concerned that this design, this case control design, wouldn't effectively capture this. But the family-based design is naturally insulated against population stratification effects because you've got families intrinsically as the unit sort of controlling for ancestry. You're measuring transmission of a haplotype, not the frequency of an allele in a population. So this approach is sort of like, family-based studies are naturally robust to these population stratification effects. So people were arguing about how much this was going to be a concern. Once you do the experiment, you're doing lots of testing. So people were imagining, okay, well, if linear Krugman is right, we have to test 500,000 SNPs. We have to induce the right multiple test correction. Is it 500,000? Is it a million SNPs? Is it 2 million SNPs? What's the right significance threshold when we're doing this sort of like at scale genome wide? We're thinking about what happens in the future when a bunch of studies actually generate data. How are we actually going to combine it over the long term? That wasn't immediately obvious. And then, the HapMap project is collecting data, but the catalog of genetic variation through other sources is sort of growing and increasing. How are we actually going to apply and perform tests when we collect larger catalogs of genetic variation? So some of these were intrinsic to, should we do these experiments? And then others of these were, okay, now that we've done them, we've got these other, issues to deal with. But it turns out that, like, by and large, most of these problems have straightforward solutions. So I'm just going to walk you through some of the key papers that sort of led to solutions to them. So starting with the first one, population stratification, there's this great paper from Alkus Price and David Reich. It came out around 2006, really just, describing this very, very simple idea that we now all take for granted, which is that you can actually use principal components analysis. So, taking the entire genome-wide data, constructing values and vectors of data, eigenvalues that summarize the full distribution of data into a finite number of axes that capture that variation in genotypes across the sample. And it turns out that that summary broadly captures population genetic similarities and differences across groups. And once you have those sort of like axes of variation that explain this sort of like broad scale, then it becomes a very, very straightforward thing to generate those values, PCA components, across all individuals, and then just include them in regression analyses to correct for that effects. And this was sort of a first paper that sort of described that idea in principle and really helped change how we think about how we think about doing this. Previous work, this is all built on previous work where the idea that you can actually use genotype data at scale to infer quite accurate estimates of population um, um, ancestry and admixture. That's work a couple years previously, but building on that, trying to summarize that and then create values that you can use in regression analysis. This is sort of like the not innovation in this particular work. Another important model that sort of came out of these around this time is this idea about modeling haplotype variation and patterns of linkage disequilibrium in this sort of like mosaic model. And this very basic model from Lee and Stevens came out in 2003. And it's a very, very amazing, simple idea that actually does a very, very good job of capturing variation in data, even though it's not a perfect model and it has some technical features that are slightly suboptimal, but actually on balance, despite that, explains data well. So the key idea of this work is that, let's imagine that you've constructed a set of reference haplotypes where you've actually gotten the haplotype phase. So the constellation of alleles that lie on that haplotype, and you've learned that relatively perfectly or close to perfectly from previous data. Okay, so the idea then is, let's say that you're presented now with a new haplotype and you want to guess at what the configuration of that new haplotype is. Well, what you can do perhaps is describe that new nth+1 haplotype from the previous n haplotypes as simply a mosaic of that previous haplotype diversity, where when you change the mosaic, the pattern in which you change from one haplotype to another is dictated by the background rate of a combination between those loci. So that's an incredibly straightforward and a simple idea. And it turns out to be an incredibly straightforward way of modeling the data. And as you start to think about it and imagining, okay, well, let's say I've got, I've created a phase haplotype reference panel of a bunch of new individuals where I've got the genotype configuration for a larger panel of genotypes than I have from the data that I'm actually trying to do my case control association, where I've only sampled a catalog or small sampling of that haplotype genetic variation. Well, if I could just know what the haplotype structure was, if I could match it back to the take my mosaic haplotype and match it back to one or more of these reference haplotypes, then it would be a simple procedure for me to make a really good guess at what the constellation of alleles on that haplotype is. It would just be going back to those references and copying the haplotype pattern and then jumping to a new haplotype only when I thought there was a recombination rate between them. And again, this is probabilistic, so you may not have perfect knowledge of what haplotype to jump to, but if you do that a lot of times, you can, with pretty high accuracy, get the correct genotype configuration in most places. And so this sort of like imputation of haplotypes and haplotype given a reference for your current set is so fundamental, where you're trying, in the statistical imputation context, you're trying to estimate the genotypes of missing variants in a study based on some phased haplotype reference. And that turns out to be incredibly important for all of our downstream studies, particularly at this time, because there are a couple of companies competing for the arrays that people were vying to buy to do our case control testing. And it turns out those companies pick different panels of SNPs to do the genotyping effort. So if you wanted to combine information across genotyping platforms, you would only be able to combine it across a handful of SNPs that overlap, which isn't genome-wide at all. So what you had to do was like go to some reference population where you had that complete spectrum of variation from both arrays in common, and then depending on what array you had, guess at all of the other genotypes and SNP genotypes in all the other sites that you haven't genotyped on your array. So that imputation procedure was really integral into bringing together different genotyping technologies that you could combine the data in straightforward ways. And this is like one model, and there are many models that play this sort of similar game, but this was a particular one that I think changed and influenced changed minds and influenced people about this problem. And so, I won't have time to go through all of these, but, each one of these challenges in turn spawned a new technology or a new statistical model that allowed us to overcome the challenges, or we used empirical data to get the right estimates of how to do or take on the problem we are approaching. And so, in a very, very short amount of time, in a couple of years, we sort of systematically faced a challenge, the community faced a challenge and devised the technology to solve it. And then we were online and able to move forward. And so, we started to do these studies and learn things. And sort of like one of the first experiments where this was done is just like an amazing result with like 96 cases and 50 controls, cases of age-related macular degeneration. So, a classically complex trait, but, it turns out actually, kind of oligogenic, not super polygenic or omnigenic. So, a handful of loci that actually matter for this trait. Where in this case, the effect size that they discovered at complement factor H, which is the association they report, had an odds ratio of like 7 or 8. And, this is a very early days of genotyping. So they weren't doing imputation. They just had a single kind of array. It was like an array of 116,000 SNPs. And it was amazing. And they had replication to show that actually this association held up. And, if you think about it by today's conventional standards, like this is a crazy experiment. It's almost impossible to believe that this actually worked at this time. But, from the point of view of hindsight. But at the time, this was sort of also crazy that this worked because we didn't have very good intuition about what we would expecting the complex trait architecture to look like. And in practice, actually, this is sort of like an example where, oh, okay, we can find this with association studies, but we probably could have also found this within linkage studies too, because the effect size is so big. So, does it end the controversy or does it continue the controversy? Okay, well, we continue to endeavor in these efforts, and many larger groups were sort of in consortium efforts were committed to this common disease, common variant hypothesis test, and a very influential group from the Wellcome Trust Case Control Consortium collected a large number of individual subjects where they phenotyped for a range of major diseases, bipolar, heart disease, autoimmune traits, type 2 diabetes, and did this genotyping effort. And, approximately this is about like 2,000 individuals for each of these diseases. They utilized a shared control design. So that allowed them to kind of like efficiently test for association in all of these traits and make discovery. And, what they found initially in this case was like this sort of 2000, case 3000 control design definitely found a very small handful of things, but not like a uniform cornucopia, a plethora of discoveries that fall out of it. But at the time, this was amazing. We went from having very little understanding of the genetic basis of disease, then studies like this definitely suggested, oh, there's absolutely things to find. And maybe what we're limited is in just statistical power because the effect size distribution might just be smaller. So what did, the Wellcome Trust did their experiment, but other groups also along the way did the same thing. So a bunch of different groups were generating their 2,000 case control sets all together, doing imputation, doing testing. And then it became clear that, oh, everyone's underpowered. So really what we ought to do is just like bring everyone's data together and perform meta-analysis. So combine the estimated effects SNP to SNP across different studies, testing a hypothesis that actually the effect size of that site is 0 versus the alternative where it's non-zero, and you're just building the case because multiple sites that demonstrate a weak effect when assembled together can generate a statistically robust effect if everyone sort of agrees that that's the effect. One of the first studies to do that is the Type 2 Diabetes Consortium named Diagram. It's not the only one. I don't know which one exactly was the first. The Cardiovascular Disease Consortium also had a group. The psychiatric disease also had groups, but this is one that I'm in particular familiar with. I was involved in these studies from one of those consortium. But the Wellcome Trust, the Diabetes Genetics Initiative, and the Fusion Study got their data together, and it turned once we assembled, we actually discovered additional loci that none of us found individually as reproducible. But these associations were incredibly robust, and all of these are still widely associated even today as some of our strongest signals that are most reproducible. In that process, at least for many traits, and again, I'm mostly familiar with type 2 diabetes, has continued at pace over the last since 2006 and today, where over time, different studies are collecting data, performing their experiments, doing that across diverse ancestries, gradually assembling, generating their data, then meta-analyzing it, generating more data, and then meta-analyzing it. And what you can see is over time, steadily we have increased the number of statistically confidently associated genome-wide associated loci, with perhaps the greatest change in discoverability coming in the most recent era, with the addition of pretty substantial increases in sample size from a larger complement of more diverse ancestry groups, but also larger sample sizes in general from DNA biobanks, which I'll discuss in a second. One good news about this is, and it didn't have to turn out this way, but a lot of us were really invested in making sure that everyone could access the kind of bounty that everyone was creating, so that it would just massively accelerate all of the downstream analyses and perhaps even lead to new kinds of technologies and tools, given the availability of the data. And in general, we've mostly figured out how to share summary statistics that emit from these data. Again, the concern, of course, is we can't, mostly we can't share individual level data in most cases, because the IRB protocols don't necessarily permit everyone to do that. But sharing summary statistics, which are de-identified, anonymous, and not connected to personal health information, that's totally on track to be shared. And modulo discussions and debates about whether or not summary data allows identifiability, which perhaps is a conversation and a topic for another time. We can share summary statistics and do that at scale, and then organize and assemble them in a single place. And for a long time, the NHGRI was trying to assemble that data together and was doing a vigilant job of doing that. But then the European Bioinformatics Institute came along to sort of help curate that process, because it has now gotten to be such a massive effort and such a massive undertaking that you really need a true dedicated bioinformatics support to create standardized summary data that can be easily indexed, organized, searchable, and available for people. And so today we live in this great time where you can go to this catalog and search for your phenotype, find all the studies, see what data is shared and what summary statistics. You can literally go to a FTP site to download, and it's an amazing time. This is not how it was in 2006, but now it is definitely how it is today. So with that data, now we can start to return to some of our more fundamental questions, the kind of the debates that we had about the components of architecture and the relationship between traits. And we were doing that with a lot of theory and a very small amount of data. But now we can reimagine those models with a lot more data, and maybe even come up with some other ideas that help us understand the distribution of data. So one of the most obvious things that I think falls out of this sort of like larger scale GWAS endeavor is this very, very simple observation that actually GWAS variants that we think are tagged with bona fide association signals for traits, we often find those signals in non-coding regions of genome. In contrast, not to say that we don't find signals in the coding genome, we absolutely do. There are absolutely tagged variants that are coding. Those are some of the most easy to access, easy to understand, functionally able, and maybe most therapeutically actionable. But that is a minority of associations that we've discovered. Most of them fall in the non-coding region of the genome, don't obviously have a tag variant that's capturing coding variation. So what it suggests and implies is that actually regulatory biology is just fundamental to understanding the phenotypic variability in human traits. And so that sort of spawns a completely different set of thinking and approaches to try to understand this. I don't know if Eric, I think Eric Lander said this. I don't know if he came up with it or he got up with someone else, but I think someone asked, a reporter or journal, asked like a bunch of thought leaders to describe their work in like 6 words or less, and I think what he said was like, human genome, bought the book, hard to read. So not 6 words, but 8 words. And I think that pretty much captures it. Reading the non-coding genome is actually quite hard. And prior to this, we've done a good job of collecting DNA sequence variation, but collecting information about gene regulatory biology that sort of like is spawned and motivated by the central observation, as well as a litany of others from other motivators from model systems and other places. A technology that also got spawned from this is the idea of leveraging the patterns of LD and the summary statistics that were generated that map to those patterns of LD to do some more sophisticated statistical analyses. So one of the more, there are lots of these cases throughout the literature, but, sort of one of my favorites is this idea of conditional or joint multi-SNP analysis that you can perform if you have a pattern, if you know the patterns of linkage disequilibrium and you can model that LD in sort of a matrix form, and you have the summary data measured from z-scores that generate and are mapping to that LD reference, then it's possible with some clever matrix algebra and some statistical trickery that you can actually do some versions of conditional analysis where you re-investigate the association of data conditional on a given SNP in the data using solely the summary statistics and that LD reference accordingly. And that's actually pretty important and transformative because prior to this, in order to do conditional analysis, you have to do that at the individual level. And there are great papers where people had actually done that. But you're working with individual level data and you're working on a single cohort of individual level data to do this. And that actually turns out to be both really hard to do if you want to learn something, because statistically, you might not have power in an individual cohort to do this kind of experiment. You really kind of need like all of the data to do it. Then you have to get individual analysts to do that and then share their data. And it's really, really challenging feasibly, technically to get this done. So this is one approach that really like leverages that and develops statistical infrastructure to make that argument, and actually works exceedingly well proportional to how well your LD reference is and how well it matches the summary data that you're giving it. And then once you have those technologies, you can actually go back to thinking about those very, very first questions we had about heritability estimation, right? Like we motivated the genetic efforts to say, oh, we should study trait X, Y, and Z because we think those traits are heritable. And so maybe we should embark on the genetic study. So now that we have embarked on the genetic study and we have perhaps assessed a particular aspect of genetic variation, let's say common genetic variation, and we want to really kind of like evaluate the common disease, common variant hypothesis formally, we might want to ask the question, like how much heritability is explained by common genetic variation? So it turns out that actually you can, with some statistical savvy, it's very, very straightforward also to develop models which perform that heritability estimation given the common genetic variation that you've surveyed. And it turns out, by and large, common variation does explain a large fraction of heritability. I think this sort of experiment also spawned a sub-debate that happened in the field and still is kind of actively discussing. Common variation by these measures doesn't, in all phenotypes in all cases, explain all of the heritability. And so this gap between total heritability as estimated from family studies or other studies and heritability you estimate from genetic variation or an aspect of genetic variation that's not the entire panel of genetic variation, so maybe common variation, maybe not all of it. So you have this gap of missing heritability, and that's led to a lot of speculation and theory and simulations and empirical practice to try to answer the question, what is the missing heritability gap? That's still an active debate, but I think we've kind of gravitated towards genetic variation does explain a lot. And as we increasingly penetrate and are able to evaluate low frequency variants and perhaps rare variants, that's going to explain an even larger fraction of that heritability. And maybe the additional leftover probably is like gene environment and other sources of genetic variation that we haven't been able to capture very readily in these designs. Another really fundamental statistical model that came out of this is the concept of LD score regression. And so basically, this is a fantastic paper, and I highly recommend everyone read this paper. But the basic idea is that actually under this very, very simple model, you can model the expected summary statistic, the chi-square test you would get from association, given a residual amount of LD that variant carries. And let's call the residual amount of LD sort of like in a window around that variant, the sort of like total R squared between it and all other SNPs in sort of like a local regional interval. So sort of like if you're in a like recombination cold spot, you're going to have a lot of LD around you. That will confound in some ways what your chi-squared statistic is, because you might be capturing some multiple genetic variants sort of like in that haplotype block. But you can model that expected distribution given that score and a couple of other population genetic parameters, population structure, and sample size. And with this model, you might think, oh, well, what could you do? What is this model useful for? Turns out it's useful for a lot of stuff. So for example, it turns out that if you make that model and you model the distribution of association statistics, you can actually, from those that slope and change in LD association by counting for LD score, you can actually get an estimate of heritability from that study. You can also start doing things like comparing and contrasting the genetic correlation between traits once you have that information. And it all just comes back to this very, very basic statistic of LD score and the idea you could just summarize the complete spectrum of the matrix of all pairwise LD with a single measurement per SNP. That's just like a transformative idea, extremely popular. It has strengths and weaknesses for sure, but definitely a good idea. And sort of what that spawned is sort of like, again, thinking not just about a single trait, but trying to understand the relationship structure between all traits, how different traits are correlated to each other. And, once you start thinking about pleiotropy, you start, there's like this spawns this whole sort of like space of trying to relate different traits to each other. And whether or not, what the causes of those relationships are. Is it mediation or is it pleiotropy horizontal? But this is sort of like empirically, you can say, oh, I've got data, I've measured scores for association, I know the relative LD-ness of those variants, adjusting for that LD, how often is the correlation of my statistics with one another. And you can see on this plot that there's just like traits that intuitively are strongly correlated fall out of this. So, epidemiologically, we know the values for triglyceride and HDL are correlated. Well, it turns out they're also genetically negatively correlated, which makes perfect sense, fits the data. Things like BMI and type 2 diabetes, we know obesity is a causal risk factor for diabetes. So some of the genetic basis for type 2 diabetes has to be through obesity pathways and measurements, and that is undoubtedly true. And so you can look at this list and just find tons of things that make absolutely a lot of sense, and then a lot of things that you might not have necessarily expected to, because you now have a large-scale phenotypic reservoir to compare a bunch of association statistics just simply by capturing patterns of LD and summarize those in neat and interesting ways. One of my favorite kind of versions of this experiment in thinking about pleiotropy is Mendelian randomization. And this is a very interesting and nice idea where, if you think about genetic variation and its relationship to phenotype, you could imagine that genotype actually sort of in the population genetic natural context, alleles are being randomly allocated to people. There's not, barring selection and structure and some weird forms of somatic variation or whatever, people are getting across the population get random dosages of copies of 0, 1, or 2 copies of an allele. Now, if we were clever and we knew which collection of variants actually relate to some exposure of interest, and like LDL cholesterol or obesity, and we wanted to test the hypothesis, does BMI cause heart disease or does BMI cause type 2 diabetes? We're really interested in generating that causal effect estimate, sort of like that linear arrow between exposure and outcome. Well, the genetic variant, if in some ways you could view as sort of like a natural perturbation. It's an instrument that nature has like randomly interjected that you can then use to actually obtain unconfounded estimates of that causal effect. And the intuition for this, if you think about it, comes from like a model that you could imagine from a randomized controlled trial design where, in a randomized controlled trial, you've got a population of individuals, you separate them into 2 groups that are indistinguishable phenotypically from one another, one of which you give a treatment where it perturbs an exposure, and then the other gets standard of care where they don't have that perturbation, and then you monitor over time what the outcomes are for group 1 and group 2, and you're hoping that group 1 lowers their exposure mediated by the perturbation, the therapeutic perturbation, and then that has a downstream consequence on lowering the risk to some endpoint they're interested in. So giving them a statin lowers their LDL cholesterol, and that tests the hypothesis, does lowering LDL cholesterol lower heart disease risk? You can do the same thing. You're now imagining the SNP as the perturbation. That's the sort of like putative drug that you're giving people, and that's been allocated to a population at random. So you can sort of use that SNP to generate this sort of effect size estimate, and not just use one SNP, you can actually use a handful of SNPs to generate that estimated effect. So you can use this idea of sort of like an instrumentally variable weighted change in the SNP on exposure to the change in the outcome. The slope of that regression under some very simple assumptions is an effect estimate of that causal relationship between exposure and outcome estimated from genetics. Now, Mendelian randomization is a super popular, it's taken over almost too much by some measures from some people. Definitely has strengths and limitations. It comes with some assumptions about needing this instrument to directly relate to the exposure of interest. You're picking SNPs that are strongly associated with that exposure. They're not obviously associated with extant confounders. And they do the effect of the SNP from the exposure to the outcome happens only through the exposure to the outcome, not sort of like an end around directly to the outcome. There are other assumptions that need to be held as well, but these are the main ones. That may or may not hold in all cases, but the idea that you can generate causality or estimates of causality from genetic variation is a powerful idea, and sort of follows now that we have this great reservoir of genetic variation associated with a range of both exposure traits we're interested in and outcome traits as well. Another thing that comes out of this is this sort of like idea of polygenic risk score. So, returning to the question, can we actually predict a trait given the genetic variation that we observe, is just this like fundamental idea that could have transformative potential, because we want to know, at an early stage before the presence of disease starts to manifest, we'd really like to know who's the most genetically susceptible so that we could perhaps early in history, in patient life history, do things that sort of like push them on the path to staving off disease or prolonging disease as long as possible, or perhaps even avoiding it entirely with the right therapeutics. So genetic risk scores and polygenic risk scores are very, very easy to calculate. The score that you can imagine for a given patient, you can model, again, in this sort of additive model, where you just, for every important site that relates to that trait, so site J1 to M sites in the genome, these are like perhaps your genome-wide associated sites. You go to the individual and you count how many alleles they have at that site, 0, 1, or 2 copies, and then you just multiply that by the effect size estimate of that allele on the trait you're interested in measuring, and you sum those across all loci, and you can get a polygenic score. And that score, not surprisingly, is sort of like relatively normally distributed in a population. And if in the sort of like perfect model where all variation is explained, is additive, and you had every genetic variant that explained every component of that genetic variation, you could perfectly capture the genetic component of variation to the trait and then use that to sort of like suss out and stratify individuals. And here's like a great relatively recent paper, which sort of like pushes this to sort of like a potentially clinically valuable insight, where sort of like the essential idea is construct your polygenic risk score. There's going to be a distribution of patients in the population, but what about those patients that are in the top 5% or 1% that have sort of like the highest polygenic risk? What is their downstream risk to endpoint disease? And it turns out that you can find, those individuals may or may not carry sort of monogenic causes of disease, but in that top 5%, the risk that those individuals carry to accrue the disease is an effect size that is at least as strong, and in some cases, approximately as strong as a monogenic cause that we definitely do clinical action on and we definitely want to intervene on. So the idea that actually even if it's not a single genetic variant that's the cause of disease, that there are some individuals that just drew the bad straw and had collected a large number of very, very weak effects, but have so many of them, they have artificially high LDL cholesterol because their genetics predisposes them highly to them. And if we can use a score to figure out who those individuals are, we could actually perhaps do something in the clinic to help prevent or slow the progression of disease. We live in also a time that's amazing and exciting. We sort of live in what I'm personally labeling like the GWAS 2.0 era, and it's called the biobank era. And so if you remember that plot that I showed a couple slides back, where in type 2 diabetes genetics, individual cohorts were assembling data and we were pulling data together. And then all of a sudden, in the last 3, 5, or 6 years, that just took off precipitously. We collected larger, diverse cohorts, meta-analyzed them together, and that's just led to a preponderance of new discoveries because the power to discover is just much larger with like hundreds of thousands of subjects participating. Well, the reason for that is because of DNA biobanks. So these are places where health systems data are connected to electronic health records phenotype data, and those health systems are progressively genotyping or sequencing those subjects. So you now have pools of people who are getting routine clinical care that have donated their time and their genetics for further research study, and the health systems are collecting those genetics, and they're doing so at scale. So tens of thousands, hundreds of thousands of people. Early on, those are pioneers like BioView and maybe to a lesser extent Decode. But UK Biobank is an easy is a very early adopter in the early days that now everybody uses. But there's just like tons of biobanks now available, and these are just great resources because they often are more representative of national demographics where they're collected. Rather than particular ascertainment of who the investigator wants to study. They have electronic health records, so there's deep phenotyping attached with them. They're profiled with genetic data, but maybe also not limited to genetic data, maybe functional genomics data. And there's a larger runway of potential additional data that can be collected in these subjects from imaging data, prescription drug use, lifestyle, behavioral data from surveys. And so there's just like a ton of potential energy from these resources from both genetic studies and non-genetic studies alike. They are biobanks, and so there are different strengths and weaknesses, and of course, different biases that are going to come from these that we're only just starting to learn about as we use these more robustly. But by and large, this data collection has substantially improved our power for discovery. And one of the things that I think this environment leads to is historically we've been thinking, oh, we should test one trait at a time, and everyone's testing their kind of interesting trait of interest. But once you have a phenotypic repository where you've measured all of the phenotypes by billing codes, or you use billing codes plus text mining or some other resources to generate large catalogs of phenotypes for diabetes and others, and you've uniformly phenotyped an entire biobank scale, you start to think, well, why do I have to just analyze one trait? Why don't I just analyze them all and try to find particular associations for that? So that introduces the concept of PheWAS, demonstrating that's feasible, adding additional layers of statistical burden, but a natural thing to do, which sort of then leads to larger problems of scale, which maybe I'll talk to in a second lecture down the road. But this is an opportunity to both fully mine and leverage the data and really capture and try to understand phenotype correlation in a biobank system. But this new era doesn't lead to like a panacea without any problems. Of course, as we're thinking about designs and trying to do particular experiments, and we're thinking about phenotyping, the data is not perfect. So like here's one example where in a biobank, you might have a very, very few number of cases tested against a large number of controls. And actually that imbalance is not necessarily expected as the right expected properties of our like common variant tests where we have an equal number of cases and controls contributing. And so that unbalance might actually lead to statistical issues, and that might require us to develop new statistical procedures to sort of adjust for that, whilst also integrating the most advanced ways of handling all the other problems that we had previously about relatedness and population stratification all collectively. And so the new types of data spur new statistical innovations and technology that then get deployed on scale so that we can share data and combine information and advance the art. Okay, so, but at this point now, we have done these experiments, we successfully mapped complex trait variation. Okay, great. Like, what do we do with this? Okay, so, there are just like a couple of fundamental lessons that I think we've learned and I've sort of impressed in the last couple of slides. But basically, a couple of things that are very straightforward immediately is that because non-coding, variants that we find are non-coding, it probably means that regulatory function matters. And there's lots of reasons for that because variation is associated with change in gene expression are found in regions where chromatin is open, as we've collected data to capture and model functional genomic variation. We find that regulatory biology in different ways is definitely implicated. And so now we start to think, oh, we should start to think about how we can use that data to understand variation more. But there's also complications when we start to think about regulatory biology. So, genome biology and the way in which genes and regulatory elements are structured are very complicated. It's not like there's a promoter element and then the 5 regulatory factors are like just upstream of that promoter element. Distal enhancer elements like are a thing, and they can connect to genes that are not the nearest gene, that are genes that are actually much further away. Sometimes they impact genes that are right nearby, but maybe actually the process that we use to sort of like call a gene may not be the right way. Like maybe the gene most likely to be associated with disease may or may not be the nearest gene. So maybe we need to start thinking about how to catalog these sort of enhancer elements, like bringing those things together and developing new technologies to make those assessments, and then projecting them onto the association data to get the right variant to gene, element to variant connections in play. In addition to that, one of the fundamental weaknesses of this LD-based design is that we have these blocks of LD where we've tested for association with one variant, and we found a bunch of variants in strong or partial LD with that variant. So you often will find and hopefully find that actually you have a variant and there's like tens or hundreds of variants that are in partial LD with that site. So that tag variant we've selected may just by chance be the most associated, but it may or may not actually be the causal variant. And probably in practice, it's often not the causal variant. So trying to suss out using the statistics alone what the right variant is, or trying to deduce what the causal variant is just from the statistical association data, is hard, maybe even impossible. On its own, maybe you can narrow on a set of variants that you think are statistically the most likely to harbor the causal variant, one or more causal variants, but you probably need other types of techniques to deduce and suss out what that causal variant is, as well as providing calibration and measurements that sort of narrow on that credible set of variants more in a very straightforward and robust way, that don't just do it with one signal regionally, that maybe actually look at multiple signals in a region to capture allelic heterogeneity, that other aspect of genetic architecture that we don't fully understand. So post GWAS, we're kind of left with these like salient questions, like what actually is the gene? GWAS was a great approach. It definitely found loci in the genome that we know statistically associate with disease beyond reasonable doubt, but it doesn't necessarily tell us what the gene is. It may not obviously tell us what the causal variant is. It may not tell us what the direction of the SNP is with respect to the gene and how that relates to the phenotypic association. Is it an increase in expression or a decrease in expression of the salient gene that's phenotypically relevant? How sensitive the system is to perturbation of the gene. Is this a small effect size variant that has a small effect on the gene? Is it a big effect size variant that impacts the gene a lot? What's the horsepower of change in phenotype, change in molecular phenotype to change in physiological trait? And ultimately, what is the mechanism? There's lots of ways non-coding variants could, in fact, modify or modulate changing regulation of that steps with one or more genes nearby. But what is actually the function that's acting downstream? And again, just like in the previous case, there are these great questions that now are spawning a great many number of both statistical approaches that try to tackle that problem, but also new types of experimental data that are trying to bring new data and insight to being to bear on those data that then will spawn the development of new methods to try to understand that. And I can't talk about all these things here, and in some of the other lectures that you'll probably hear more about many of these. And so with that, I'll stop and tune in a little bit, and I'll kind of go over a little more detail, some future thinking and some contextualization of some of these ideas and sort of thinking about the future. But thanks for listening.